Hey, I'm Dr. Christopher Pritchett in the Department of Health Sciences at East Tennessee State University. And today I want to talk a little bit about vaccines and how they work. And so I've made this little PowerPoint slide to sort of go over some of the important aspects of vaccines and to give you a little bit of an idea of how vaccines work. So there have been two major developments in human history that have allowed our life expectancy to increase. And the first of those is the development of a clean water supply. And the second of those was the development of vaccines. And the development of vaccines is something that for a long time we really weren't sure how vaccines were protective. And now are getting much better at understanding this. And vaccines have eradicated several diseases, including things like smallpox. And in the US, this is why we don't have polio anymore. And so there have been a lot of great benefits and reduction in morbidity and mortality around the world because of the use of vaccines. So vaccines are one of these things that are absolutely crucial to a healthy society. And so I want to talk a little bit about the different types of vaccines and how they work. And in order to talk about vaccines and how they work, we have to talk a little bit about the immune system. And so the immune system of humans, we sort of break up into two sections, the innate and the adaptive or acquired immune system. And shown on this slide over here on the left is the innate immune system, which is made up of both physical and chemical barriers to infectious agents. So this would be things like your intact skin or the acidity in your stomach and so forth, mucus, things like that. And then you also have cells that function as part of your innate immune system. These could be cells like this dendritic cell here or this macrophage that are like little Pac-Men that go around and ingest anything that you might have been exposed to that you're not supposed to have in your tissue. Say, for instance, if you get a splinter or something. And the innate immune cells, though, are really critical because one of the things that they do is they stimulate the process of inflammation. So inflammation is a normal response of our body. And if it's an acute and it's local, then that's usually a good thing. If it's chronic and systemic, then that's a bad thing. So you don't want systemic or chronic inflammation. You want inflammation to be controlled and be local and acute. You want it to resolve at some point. And so what happens during this process of inflammation is that many of your cells of the innate immune system start to communicate with other cells. And these are the cells of your adaptive immune system, which are your B and T cells. So shown here is a T cell and a B cell. And so these B and T cells are something that we develop throughout our lifetime. And they're important because they have this ability to specifically recognize individual pathogens and individual agents that we might potentially be infected with. And so these B and T cells have to get this information from your innate immune cells in order to function properly. So there's a whole lot of chemical crosstalk that's required for your adaptive immune system to get up and running. And so B and T cells are really important because they have this specificity to them. They are going to specifically recognize only one certain thing. And because of that, one of the things that happens during the development of an adaptive immune system where you've activated some of these B and T cells is that some of these cells will then further develop into what are called memory cells, which they have shown over here on the far right. And these memory B or T cells are what are so critical to understanding how vaccines work. So the goal of a vaccine is to stimulate the production of B and T cells that are going to specifically recognize a particular pathogen and provide protection to you so that after you get vaccinated, if you're ever exposed in the future to this pathogen, that you will produce these specific B and T cells that specifically attack that invading pathogen and prevent you from getting sick and also prevent you from spreading the disease throughout the population. And so these B and T cells can do a variety of things. And the one that you've probably heard the most about are B cells that develop into these plasma cells and start secreting all of these antibody molecules, which are these Y-shaped structures that you see being secreted by this B cell. In some cases, the T cells can do one of two things. In some cases, the T cells become these killer cells, which they basically run around and target specific cells that have been infected by a particular pathogen, whether it be a virus or a bacteria. In other cases, T cells will act as sort of a referee and determine how the immune system is supposed to respond to a pathogen, a particular pathogen. And so really, in order for vaccines to work means you have to have this adaptive immune system. And that also means that you have to have this crosstalk between the innate immune system and the adaptive immune system. And this happens in a variety of tissues, but especially it happens in the lymph nodes. So if you remember the lymph nodes, when your parents said your glands were swollen, those were lymph nodes. So our tonsils are actually a lymphoid organ as well. And then we have lymph nodes various places in our groin and in our armpits and behind our knees. And they're in a variety of places anatomically throughout the human body. And so a lot of this talk between the innate and the adaptive immune system is critical in order for vaccines to work well. And so the basis of vaccination is to protect the society at large. And in order to protect the society at large is something that's been in the news quite a bit recently, which you've probably heard about, called herd immunity. And so herd immunity is where there's a number of individuals within the population that have been vaccinated. And because there are so many people who have been vaccinated, this protects people who haven't yet been vaccinated against a potential infection. And the one thing that hasn't been quite clear is that every infectious agent has a different sort of threshold for how many individuals within a population has to be vaccinated. And so this concept of herd immunity working requires a large percentage of the population to be vaccinated. And that's sort of illustrated here in this slide. So you can see these orange cartoons here are the patients that are infected. And if you only have a few people in the population that have been vaccinated, such as here in blue, this leads a large number of susceptible individuals, these gray people that you see, these gray cartoon structures, that can now be infected by this infected person here. And so you can see what happens if you don't have enough of the population vaccinated, you end up with a lot of people getting infected. And that may be something as simple as the common cold, or it could be something much, much worse in the case of SARS-CoV-2. And so if you get enough of the population, and so the reason that all of this talk has been about developing a SARS-CoV-2 vaccine is because if you can get enough of the population vaccinated, then even if you have a few people who are infected, they have a much less chance of infecting the majority of the population, because many of these gray cartoon structures, as you can see here, are actually protected from infection because they don't have a chance to encounter that infectious agent that's being carried by these infected patients. And so that's really the basis for herd immunity. And that's going to differ for every infectious agent. And it's also going to be determined by the population as well. And so herd immunity is really the basis for, and probably the most important concept of understanding how vaccines are important as a public health measure to protect the population. And so there are different types of vaccines that can be given out there. And so I wanted to talk a little bit about this in terms of the SARS-CoV-2 and in terms of the historical perspective, because the development of these vaccines is really been crucial to our establishing our lifespans. And so in the case of passive immunity, this is a short term. This doesn't generate active B and T cells like what we talked about at the beginning. And so passive immunity is something that you get when you get antibodies delivered into your body to protect you from an infection. And this occurs naturally when we're in the fetal stage inside of our mother, where antibodies are transported across the placenta to the fetus. And these antibodies actually persist within the newborn for several months after birth. And these protect the infant from exposure to a variety of infectious agents. And over time, those antibodies go away, and then the newborn develops their own adaptive or acquired immunity due to the development of B and T cells, and then they're no longer protected by those maternal antibodies. So passive immunity doesn't last as long. It doesn't develop that memory response. And you're also capable of getting a passive immunity artificially. And so this would be when you get convalescent sera, for instance, in the case of some SARS-CoV-2 patients, getting convalescent sera from other people who have survived to lessen or decrease the severity of their symptoms. So this would be another example of artificial passive immunity. And so the other classic example of this was treatment for snake bites using antivenom, which were basically antibodies that were derived from horse serum that were injected into people to protect them against getting dying from snake bites. And so, passive immunity is a good thing if it works. The problem is that because it doesn't generate a long-term immune response, it doesn't activate those B and T cells. The passive immunity doesn't result in you being protected from future exposure to the infectious agent. And so the development of vaccines is really a critical aspect because, as you can see over here, this leads to the activation of your B and T cells. So really the difference between active immunity and passive immunity is the ability of those treatments to activate your B and T cells. And you can develop active immunity either by getting exposed to the pathogen itself and recovering and then having that convalescent sera, which then could be used in a passive immunity scheme over here, as we talked about. But the other way of developing active immunity, which is the basis for vaccines, is to activate those B and T cells before you get exposed to that infectious agent. And that's really the goal of vaccines, is to try to prevent you from ever getting sick and being contagious or developing severe symptoms due to that particular pathogen. And so vaccination has really been one of the major foundations of human society. And so active immunity can come about by a variety of different vaccines. And I just wanted to mention a few of the different types of vaccines that are out there. And so this slide nicely lists the pros and cons of some of these types of vaccines that are available out there. So one of the ways we can make a vaccine is by inhibiting the ability of that pathogen to replicate. So, for instance, the flu mist vaccine is an attenuated virus. So this is a form of the flu virus that can only replicate at cold temperatures. And so what happens is that when you get the flu mist vaccine, it only replicates in the upper nasal passages. It doesn't actually get into your lungs. And because it's cold adapted, it won't grow at normal body temperature. But there's always a safety issue with the live attenuated vaccines. And so people try to steer away from them if possible. And so the other type of vaccine is where you take the pathogen and you completely inactivate it. So in some cases, this is by treatment with very harsh chemicals or what have you. The thing about this is that while it works well and it's a very safe vaccine, the problem is that it doesn't always activate your B and T cells to the point where it should. Whereas the live attenuated vaccine usually activates your B and T cells really well, but there's always a safety issue with that, because if you have some of this replicating pathogen, there's always the potential for reversion. One of the safest vaccines that are out there are like the subunit vaccines. And so like the hepatitis B vaccine that's given to especially healthcare workers. And this relies on purified proteins. So you've eliminated any aspect of or potential for a pathogen to replicate. And this is actually purified protein that's injected with something we call an adjuvant, which stimulates your inflammatory response. And so you develop these B and T cells to these proteins that are injected that have been purified. And so these they don't have any nucleic acid, so they're non-replicating. And the good thing about subunit vaccines is that they are extremely safe, because there's no potential for reversion. There's no potential for developing any kind of resistance or anything like that, because these are just biological molecules that can't replicate by themselves. The problem with subunit vaccines is that they oftentimes don't last as long as, say, these other two, the attenuated or the inactivated vaccine. And part of the reason from that stems from the ability of the subunit vaccines, the proteins that are injected into you, to be maintained within the tissue and within the body to generate enough memory B and T cells so that you're protected for a very long time. So, for instance, this is why you frequently have to get booster shots. So the boosters are there to give you another injection of these proteins so that you keep stimulating your immune system to make sure you have a good B and T cells made to that specific pathogen. So these would be things like the diphtheria toxoid. So a lot of the toxin-mediated diseases like pertussis and diphtheria and tetanus, those are inactivated toxins that no longer have any function and they can't work the way they're supposed to as a toxin. And so the reason you have to get a booster is because your immune system doesn't develop a good active memory response. And the advent of molecular biotechnology in the late 70s has allowed this new group of vaccines to be developed, some of which are based on nucleic acids, so using DNA molecules or RNA molecules. And the nice thing about this is that they're usually very safe, but the problem with them is that there's only a few that have actually made it to the market. And so you've probably heard about Moderna's SARS-CoV-2 vaccine that's actually made it into clinical trials. And that's an RNA vaccine. There is actually a vaccine for horses that's a DNA vaccine. So the nucleic acid-based vaccines are something that are relatively new, and people are really excited about them because this is a new way, basically, of activating our B and T cells. And then there are a variety of other methods that we can make all of these, what we call chimeras, to develop an active B and T cell system. And so there's a whole variety of these out there. And the other part of this slide, this viral-like particles that's pictured over here, would be indicative of things like the papillomavirus, the HPV vaccine that you've probably heard about. So the nice thing about these, the viral-like particles, the recombinant viral vectors, and the nucleic acid and the subunit vaccines are that these usually don't replicate in the human, and so these usually have pretty good safety. The biggest problem with these vaccines is the public perception. And so it's important to understand the difference between these different types of vaccines that are out there, because these vaccines that have been derived since biotechnology, this molecular biotechnology revolution, this recombinant DNA technology, now eliminates a lot of the hazards that were present for vaccines in the past that used these attenuated or inactivated vaccine candidates. And so the development of biotechnology has really allowed us to come a long way in vaccination. And so you're going to see more of the SARS-CoV-2 virus vaccines that are being developed that use these recombinant methods. And so the first one is the RNA vaccine from Moderna. But there are several others in the works. And they've also, as I mentioned, used the convalescent sera. But there are several other vaccines on the horizon, some using these viral-like particles, using some of the spike protein from the SARS-CoV-2 virus. So there's a variety of vaccine potentials out there that are being developed currently during the SARS-CoV-2 pandemic. So I hope this helps to explain a little bit of vaccines and a little bit about how they work and a little bit about your immune system. Thanks.